Hello, today I'm going to talk a little bit about the article titled Gal4 System in Drosophila, a Fly Geneticist Swiss Army Knife. This article was published in 2002 and was written by Joseph B. Duffy from the Department of Biology at Indiana University. Duffy aims to examine the Gal4 UAS system. He first defines this system and then goes into the various ways that you can use this smart genetic tool, depending on exactly how you wish to analyze gene function. I want to briefly explain the Gal4 UAS system as simply as possible. So, the Gal4 is a gene regulator found in yeast that can bind to certain base pair sites between Gal10 and Gal1. The base pair sites are the upstream activating sequences, aka UAS. The sequence is like an enhancer element for the Gal4 regulated gene transcription. So this biochemical method takes advantage of the relationship between Gal4 and UAS, and because there's an activation of transcription that leads to control over gene expression, scientists can identify the function of certain cells or genes, which is quite revolutionary. The article goes over many experiments to convey the effectiveness of this system, and I'm going to be talking a bit about the first one, which is depicted in Figure 1. The experiment shown in this figure was performed by two Harvard medical scientists called Brand and Perrimon. They were interested in controlling cells and their phenotypes through targeted gene expression. Brand and Perrimon are mentioned multiple times throughout our article because they did numerous different studies with the Gal4 UAS system, but the one Figure 1 is describing refers to a study they published in 1993 called Targeted Gene Expression as a Means of Altering Cell Fates and Generating Dominant Phenotypes. Once it was discovered in 1988 that the expression of Gal4 would lead to the transcription of a reporter gene under the control of UAS, Brand and Perrimon decided to look into this with Drosophila and see if they could control and manipulate cells and generate dominant phenotypes. And they took what the article refers to as a bipartite approach, meaning that there are two essential components in this system. And in this case, it's two separate parental lines. One parental line is coined the term responder, and in this experiment performed by Brand and Perrimon, the responder is the female fruit fly. And then in the second parental line, they use a male fruit fly, and they refer to the male fruit fly as the driver. The female fruit fly carries the UAS responder, as well as a green fluorescent protein, and the male fruit fly carries the Gal4 driver. Since the female responder lacks the UAS, it does not express the gene of interest, which in this case is the GFP. Therefore, the male and female Drosophila are mated to activate transcription of the GFP. This would result in different progeny containing all the elements, and the recombinant would express the GFP. So this experiment makes it evident that the system is effective in studying toxic or lethal genes due to the initial transcriptional inactivity in the responder gene. And Brand and Persimon use this versatile system for studying oncogene genes and loss of specific cells, making it very clear that this system can be used in a variety of different ways. While Joseph Duffy's article may seem a bit outdated as it was written in 2002, and of course the experiments referenced are even older, the experiment I went over was published in 1993, it is regardless still an in-depth look into the Gal4 system, which is still incredibly relevant today, and is also something we should understand as much as we possibly can, because we will be doing it in lab this semester. So while this article is simply a review in some sense of things we will be doing and learning this semester, the article further bolsters how this biochemical tool can make the study of human genes more feasible, as the many adaptations with Drosophilas that have been done in the past have proven to reveal a lot of information.